Welcome everybody to this first online webinar of the Young Investigators Committee. And I'm happy to see so many from both, or all continents actually. Today we have Professor Joris Rotmans and as our first speaker, and Arvin, Soepriatna and myself will chair this session. And Arvin will give an official opening. So thank you for joining and yeah, have fun.
Yeah, thank you again for everyone for attending this first ISACB webinar series. We're very excited to have everyone from across the globe and across multiple time zones, as Merle said. In fact, I believe we have participants from over 10 different countries for this webinar, which really highlights the tremendous impact that the work of our invited speaker, Dr. Joris Rotmans, has. So before we begin, I would like to make a few statements about the webinar format. Dr. Rotmans will give a 45-minute to an hour-long presentation, and there's going to be a discussion at the end. But if you have any questions, feel free to ask them during the presentation. In fact, we do encourage this instead of waiting until the end to ask the questions. If your bandwidth allows, we prefer that you leave your camera on. And then when you have a question, please use the raise hand icon on the Zoom, and we can unmute you so that you can ask your question. Alternatively, you could also write the questions in the chat that Merle and I will keep an eye out, and then we'll restate those questions as the presentation goes. So it's our pleasure to introduce our first speaker. Dr. Joris Rotmans received his MD and PhD degrees from the University of Amsterdam, where he studied novel therapeutic strategies for improving vascular access in hemodialysis patients. He then completed his postdoctoral training on vascular tissue engineering from the Australian Institute of Bioengineering and Nanotechnology. He is currently a professor of internal medicine and the head of the nephrology medical residency program at Leiden University Medical Center in the Netherlands. As a physician scientist, he combines his clinical duties with his research, which is funded by a highly prestigious BD grant from the NWO. He has over 120 publications focusing on various aspects of kidney disease, including the development of a tissue-engineered blood vessel for vascular access. Finally, he is a president-elect of the Vascular Access Society and chair of a vascular tissue engineering focused Hermes group. Today, Dr. Rotmans will give a broad overview of his research and how to translate vascular in situ tissue engineering from the bench to the bedside. And with that, we'll hear from Dr. Rotmans with his exciting presentation.
Well, thank you very much, Arvin and Merle, for this kind and extensive introduction. For me, it's really a pleasure to be here today in this first webinar, and I really like the setup where we have quite some time, in total 1.5 hours to discuss the topic of vascular tissue engineering. And indeed, I will give a presentation. So how often are you allowed to present for 45 minutes? Not so often on normal conferences. But on the other hand, my goal is to make it as interactive as possible, right? Because these webinars, especially with these relatively small groups, are very suitable to interact and to exchange ideas, not solely about the content of the research, but also about more general issues about translational medicine, how to bring technology to the clinic and so on. And that's, at least from my perspective, a second topic apart from the scientific content that's worthwhile discussing today. So I also included some questions and polls for you in my presentation, and I would highly recommend you to anonymously, which is possible, provide some input to these questions, and well, we'll see how that goes. And so please use the option to ask your questions that may raise during my presentation or that you had in advance. And so I'll start with sharing my screen and see if that works. And well, before I actually start my own presentation, I would like to highly recommend you to all visit the 18th biannual meeting of the ISACB in Memphis this year, from 28th of September to the 1st of October. Especially if you like music, you should definitely go to Memphis, of course, but also apart from those cultural issues, I think this is a very good meeting to interact. Especially after this COVID time, it's very encouraging to meet people live and to exchange ideas and to build on your network. And that could be also a good opportunity if you're not already to become a member, right? Which also provides option to interact with your colleagues and to build a network for future studies. Because as you probably all know, collaborations and interaction is the way forward to, especially in this field, which is very complex and multidisciplinary. So forget about the ideas to really change the world on your own. You really need others for that, and societies like the ISACB are very helpful for that. So that's what I wanted to start with. And then I assume that you can see my slide, my first presentation. As I already was mentioned, I'm an nephrologist at Leiden University Medical Center in the Netherlands. And there I'm working as a physician, combining the treatment of patients with kidney disease, so patients on dialysis, but also transplant recipients, and research. The research part is primarily focused on vascular disease. And this is a starting slide for the questions that will come across during my presentation. And this is the moment to log in to this WooClap application. You could do it by entering the address, which is depicted in blue on the top of part of the screen on your phone or on your computer, or use the code which is depicted on the left, which is a bit small, but I was not able to enlarge that. But we checked in advance and it seems to work. So hopefully you can all log in. I'll wait for a little while that you can see, you can give it a try. We can see at the bottom of the part of the screen the number of people that is logged in. And let me see how many we are with 34, so we have still some work to do. Okay, and for those who are not logged in yet, otherwise please write down the address so that you can do it a little later, so we can move on.
I will put the web link in the chat, so then you can still...
That's even better, Merle. Perfect. Okay, so I'll start with this video. Probably some of you are aware of that because it was really breaking news in the 1990s. So by Dr. Vacanti, who actually managed to grow a human ear on the back of a mouse. And I do think that was really the starting point of this, that accelerated the field of tissue engineering, because it illustrated indeed that these kind of things were possible. You could argue, of course, well, what's the need of that? And I don't think that this technology has been very useful for humans in general, but it showed indeed what could be done. And since then, a lot of progress have been made. I think in 2013, I checked recently, there was one of the first publications really about tissue engineering of ears. Well, that was of course to some extent being done with the use of. I just wanted to see if I can get rid of these upper panel in my Zoom account, but I think I'm not able. So I hope you can see the titles of my slides, because this bar of thing is covering it. There we go. Let's do it like this. Okay, and since then, various applications of tissue engineering of tissues and organs have been brought to the clinic, primarily in the years between 2005 and 2010. A lot of publications came out. For instance, on the left, you see a tissue engineered bladder that was implanted in humans. Tissue engineered heart valves. A lot of work has been done also since then. to improve these the outcomes and the performance of tissue engineered heart valves, where of course hemodynamic conditions are quite challenging, but the structure of the tissue, for instance, is not so complex as, for instance, a kidney or a brain. So the next one is tissue engineered skin, which was applied, and tissue engineered trachea, and since then, well, from all kinds of organs, attempts were being made. So a lot of successful therapies were developed and brought into clinic, but at the same time, none of these diseases, as far as I'm aware of, is really the application of tissue engineered tissues and organs really the current standard of treatment for replacement. So it's still on a very small scale. If you look to blood vessels, of course, if we have the aim to mimic blood vessels for vascular graftings in humans, of course, the starting point is to learn about the anatomy and the function of blood vessels in humans, and in this case, of arteries. And you, well, as you probably all know, as researchers in
Dr. Rotmans, you're muted.
Yeah, someone accidentally muted me, so I hope it's be undone now. Yeah, so it's about prevention of coagulation, prevention of inflammation, and the vasomotion, of course, of the blood vessel is very important, which is a function which is primarily being provided by the tunica media. And on the lower part of the slide, you can see some other characteristics that are also relevant for the application of vascular access for dialysis, which I will talk about later, which includes suture holding strength. So you should be able to suture the graft, of course, into the vasculature. It should be leak-proof, obviously, and infection resistance. It should have some compliance as close as possible to the compliance of native blood vessels. And for the application of dialysis access, to be resistant to repetitive punctioning, and it should have an adequate burst pressure, which of course is important to prevent the rupture of the vessel. And related to that topic, I made a question for you, and you can see it here. And the question is just to get some physiological input, knowing that the blood pressure of a human being is about 120/80. What's the burst pressure of a human artery in general? I'll give you some time to provide an answer. Try to make your own decision, right? And not get influenced by the bars that you already see, because the majority is sometimes wrong in life. Okay, almost getting there. 10 more participants to go. All right, so in this case, the majority is right, because the correct answer is 4. It's 1700 millimeters mercury, which is of course excessively high, because that's a condition that almost never occur in the human body. But it resulted indeed in the requirement of the burst pressure for tissue engineered blood vessels, which is also in that range. Right? So if you want to make blood vessels that are suitable for vascular grafting in humans, the burst pressure of these vessels should be in the range of 1700 millimeters mercury, which is quite a high demand, especially if you use biological scaffolds. And here you see an overview of different techniques that have been tried in the past or still are ongoing for in vitro engineering of vascular grafts, where sometimes mandrels are used, as you can see here on the top panel, where the cells were grown in seed and rolled around these mandrels. Sometimes extracellular matrix proteins or polymers were applied around the mandrels, and then subsequently the mandrel was removed. You could also use, as on the lower part of the slide, xenogenic sources of scaffolds with or without cells. And then, of course, with these xenogenic sources, do a decellularization step to get rid of these, well, mostly porcine cells. And then some technologies, which I will discuss later, use a cellularization step in the laboratory. Others choose to implant an acellular graft with the aim to get it cellularized after vascular grafting. And these are, well, just various general types of strategies that has been pursued and that I will discuss. So, but I just wanted to get some input from you, and not about, well, the ones with the best results, but if you would be a researcher and you would like to write a program for vascular tissue engineering, what would be your idea of the best scaffold to use for vascular tissue engineering? And you can see here the 5 options that I thought of, because they were used in the past. All right, so you're doing fine. Most of you already submitted an answer. Oh, I find it quite interesting that none of you actually selected a xenograft. The majority, because obviously there's no good answer, it's just a preference. The majority prefers to use human extracellular matrix scaffolds for vascular tissue engineering, and of course, to some extent, it makes sense because that probably closely, the most closely mimics, well, the normal human anatomy. Yeah, the question, of course, is how to get a human extracellular matrix scaffold or how to make it. Yeah, but this is insightful to see what you think. This is a table of a paper that was recently published in Circulation Research, and I thought I just saw the first author in the audience today, which is a good option to congratulate, to provide some congratulations with this very nice work. And this is a figure from that paper showing the pros and cons of the different types of vascular grafts that can be used for vascular grafting, of course, for different vascular diseases. So it ranges from prosthetic grafts to xenografts and cryografts, so human blood vessels that have been cryopreserved prior to vascular grafting, and tissue engineered blood vessels, no matter how they were being made. And as you can see, they all have advantages and disadvantages. And that's one of the reasons why you do see such a large variation in type of scaffolds that have been tried in the past, primarily in preclinical studies. Because then the number of methods that really ended up in clinical trials is still quite limited. And it depends also, of course, to some extent to the type of application that you primarily are interested in. Which of these characteristics depicted on the left is the most important? Because as I just mentioned, for instance, for dialysis access, where most of the tissue engineered blood vessels in patients were being used for, the ability to cannulate them is quite critical. While for peripheral bypass grafts, that's not a characteristic which is relevant at all. So, well, these are factors to take into consideration when you think about additional methods to develop tissue engineered blood vessels. And it's good to realize, and I just mentioned it already in the beginning, that to really make it successful, you really have to have a lot of knowledge about biology, about mechanics, about assembling cells into scaffolds. And the only way to become successful in this field is to really get involved into such a multidisciplinary team because without all that combined knowledge, there won't be a very good solution that is suitable for a clinical application. And just look for other people that you can find in your close environment or a little bit further away, and just see if you can find the best people that can help you out with the technique of which you feel that could be helpful to your work. So I just wanted to discuss a little bit about animal models, because that is also quite relevant in this field, I feel, because as some of you might have experienced in the past who've been doing small or large animal trials, it's quite challenging to complete those kind of studies because, well, humans are different. Some animals are different from humans, at least most of them are. These studies are quite expensive and long-lasting. And it's good to realize why you actually do these type of experiments. So do you do it for safety? Do you do it for efficacy? And as I already expected to some extent, most of you say that you do animal experiments for both, for safety as well as efficacy. Well, I do think it's still a matter of debate if you really need studies for efficacy, and if so, what are then the endpoints to meet? For instance, in the vascular field, I know for arteriovenous grafting, in the past everybody thought, well, the pigs are the best model to use for vascular applications. But for instance, if you look to a vascular graft, of a prosthetic graft, the majority of the prosthetic grafts in animal model, in pig models, occlude in 8 weeks. So if you then force yourself to show efficacy or functionality of these tissue engineered grafts in a pig model of arteriovenous grafting, that you want to show that they are durable for, let's say, 6 months, even if you put in a normal, completely healthy porcine artery, that will not be the outcome. So it is, if you want to show efficacy and convince regulators and yourself that the techniques that you're developing are safe and efficient, then it's quite critical how to define the endpoint of efficacy. Merle raised her hand. That's very good.
Yeah, so I thought maybe I could open the floor with a question about this. So you talked about the regulations. So if I remember correctly, if you want to translate such a graft or any kind of scaffold, then you have to adhere to all kinds of ISO norms that I think have also some endpoints in it, but not specifically the type of animal, right? Am I correct? Can you kind of choose and tweak still the model that you want to use for this?
Yes, as far as I know, it has not been defined clearly for, well, let's say, vascular purposes in general, that you have to do it in non-human primates or in goats or in dogs or sheep or whatever kind of animals. So you have to discuss with these regulators why you choose for a specific model, and of course, first convince yourself that the model that you choose is a proper one, and that with the endpoints that you define in discussion with them, that those endpoints are sufficient to state to make statements about safety and efficacy. But that's indeed an open discussion. The more and more people did these kinds of studies, of course, the more and more there's being referred to previous investigators that use specific models. But to give an example, in dogs, it's quite known that blood vessels do not thrombose at all. So if you implant a non-endothelialized graft in sort of medium flow conditions, so not in arteriovenous conditions, but in the arterial circulation, even though the graft might not be perfect, you probably will not see acute thrombosis in these grafts. Well, if you would implant the same blood vessels in pigs, for instance, I'm pretty sure that unless you administer a large amount of anticoagulations, these grafts will thrombose. And so from that perspective, I won't say it's a game, but it's an ongoing discussion with the regulators which model to select to convince yourself, from my perspective, primarily that the technology that you're developing or the blood vessels that you're making are better than the current standards. Like in the case of arteriovenous grafts, a conventional ePTFE graft. And for that, of course, you are limited to larger animal models because it's impossible to implant a PTFE graft in a mouse or in a rat or some tribe, but that is quite challenging. And as you can see in this overview, that gives some sort of idea about the type of animals that have been used in the past. And well, also for mechanical characteristics and risk of thrombosis, of course, the dimensions of the blood vessels are quite important. So if that answered your questions, hopefully, Merle, I had also another question for the audience, which I just want to put on the table, whether or not you feel that a successful large animal trial is needed before a first-in-man trial can be initiated, or can you also go from, well, let's say a rabbit to a human trial, or from a rat to a human trial? Yeah. So the majority, although the voting is still going on, feels that such a large animal trial is needed. And that's indeed also, in my experience, what regulators ask you to perform. Especially in the Netherlands or in Europe, it's probably the experience of some others as well. It's getting more and more difficult to actually perform animal trials because there's a very strong debate about the reduction of animal studies and looking for other alternatives, while on the other hand, the regulators are still quite strong to perform these animal trials. And personally, I also feel that the application in a large animal is critical to convince not solely the regulators, but also yourself that in these high flow, high pressure conditions with dimensions that mimic the dimensions in humans, that your blood vessel is functioning well in that condition. But if you then look to the type of animals that have been used thus far, you can actually see that the majority of the animals that have been used for tissue engineering purposes, also for vascular grafts, are rats, mouse, and rabbits, and that the number of studies and large animals that have been used is relatively limited. And also, of course, it also makes sense for the follow-up, right? Because it's impossible to do a follow-up study in a mouse of 2 years or so. And for that, you really need, if you feel that it's needed to do a 2-year follow-up study, which I doubt, to be honest, if that's really needed, and then that can only be done in these large animal trials. This is an overview from the same paper in Regenerative Medicine from this year, showing the type of the dimensions and the length of the graft, which are from a functional perspective quite important to get as close as possible to the human application. That, of course, the larger the animal, the closer the dimensions are to the human blood vessels. And there's always a debate about endothelialization in these grafts, because, of course, the endothelium plays a very important role in vascular function. And so suppose you have a graft of 1 centimeter, then still the complete graft can be endothelialized just by ingrowth of the adjacent vessels to which the graft was applied to. While if you have a graft of 6 to 10 centimeters, which is far more commonly used in humans, of course, then the chance that the graft will be completely endothelialized by that ingrowth from the surrounding vessels is much, much smaller. And if you really want to study endothelialization from circulating cells, of course, these small animals are even more challenging, or to say it more strictly, almost impossible to use to answer that question. Well, the first major publication about vascular tissue engineering is from 36 years ago, from my work in Science. And I assume that everybody involved here in this webinar today has the ambition to publish in Science or dream, but it's extremely challenging to get in there. But it was also, of course, the case in those days. But it's quite remarkable to observe that this paper ended up in Science, and I think because of the breakthrough and the technology. But as you can see, the burst strength in this of these blood vessels that were just grown in vitro using a collagen scaffold and different cell types that are normally present in blood vessels, as you can see, was less than 100 millimeters mercury. And these vessels have never been implanted in a living organism. So they will definitely be a rupture immediately, but it showed, it opened the field to work on this, on tissue engineering of blood vessels. And I think that was the primary reason why it was published in such a high impact journal already at the time. And actually the first successful application of tissue engineered blood vessels was published by Toshi Shinoka, who's still a very active cardiothoracic surgeon, a pediatric cardiothoracic surgeon, currently working and living in the United States. And he did actually very impressive, and is still doing very impressive work on vascular grafts for patients, for pediatric patients with mostly congenital disease. And he actually published this paper in the New England more than 20 years ago of a child of 4 years old, where he actually implanted a tissue engineered graft. There was a patient with pulmonary atresia, so it was actually in sort of absence of the pulmonary artery on the right side, and that has a very poor prognosis. And she already had some previous surgical procedures to see if that could be solved, but not successfully before actually this tissue engineered blood vessel was implanted. And here you see an overview of the or an example of on the left how the scaffold looks. And if you look to it now, it's an EM image of the scaffold. It looks well, relatively disorganized, if I may say so. It was a combination of polycaprolactin and polylactic acid. It was seeded with autologous cells, initially from the bone marrow and later from, I think, from subcutaneous biopsies. Was processed in 10 weeks, and now 20 years later, this was a cohort of 17 patients, and still the long-term follow-up of these grafts is still quite good. Some of these patients needed an intervention in the last 20 years, but the majority have still a functional graft. And of course, this was in the what's so-called right circulation, so right-sided circulation. So in the pulmonary artery, pressures are lower than in the systemic circulation. But still, this was a major achievement, and since then he tried to further optimize this work, and is still ongoing. Merle, you had another question, or?
Yes, sorry. I kind of wanted to link this sheet or these results to the remark you made earlier, that you said, if I quote correctly, that you couldn't really understand why it would be relevant to have an animal follow-up of more than 2 years in an in vivo study. Probably there are some exceptions to that remark, but does that mean that you think that the primary failures occur in the short term? Because for most of these patients, there really has to be growth and remodeling, and maybe at a later stage, events could take place. So would, in this case, it not be a good thing that you have large animal trials that have a bit longer duration? Or do you think they would have predicted this outcome?
Yeah, that's a good one. Well, maybe what I primarily meant is that, of course, ideally you would have very long-term follow-up studies that you use sort of, I can't find the English word for that, so very young animals that are growing and are not adult yet, and then you have follow up them for a couple of years and see that these vessels remodel. And especially for the application in children, of course, that might be needed. And but what I realize more and more is that because of all these very stringent rules, and that on the other hand, if we really force ourselves to do follow-up studies of years and years, that substantially limit the translation of potentially very powerful and innovative strategies, and that they're all get stuck in the very high demanding studies that sometimes are being tried prior to these human application. And of course, safety first. But if you really want to make sure that never ever in the development of a clinical trial a unwanted event occurs, well, then none of these therapies will actually end up in a patient. It's my feeling about that. But I agree that if you primarily make blood vessels for children that are growing, you want to see that the scaffold and the constructs that you implant have the capacity to grow, because that is a major limitation that you would like to tackle, right? Because the prosthetic grafts that are currently being used do not grow. But so, if, for instance, if I make the translation to vascular access for dialysis, where most of the blood vessels that are currently being used have a primary patency, I'll give away, that's another question, less than 50% at 1 year, then you could argue, if you really want to, if it's needed, to show functional patency 2 or 3 years after implantation, because already if the patency is lower at 1 year, you already made major progress in comparison to the current golden standard. And yeah.
Yeah, I understand. Yes.
Yeah, so that's thank you for the swift bridge to that. Brings me to the introduction of the complication or the clinical disease or the clinical challenge of vascular access for hemodialysis. So patients with chronic end-stage kidney disease, let's say less than 10% of normal kidney function, depend on renal replacement therapy, as you might know. And there are various options for that. Dialysis, where you have peritoneal dialysis, where you use the peritoneal membrane as a semi-permeable membrane to get rid of toxins and fluids, or hemodialysis, where an extracorporeal artificial kidney purifies the blood. And that's what this is about, because you need a vascular access to transport the blood to the dialysis machine, and after the dialysis machine did its work, bring the blood back to the patient. And obviously the third treatment modality for patients with end-stage kidney disease is kidney transplantations, which in general is the best, but because of the shortage of organ donors, and sometimes also the comorbidity of patients, especially in the elderly with a lot of vascular and cardiac comorbidities, they were just not suitable for receiving a kidney transplant, including the massive amount of immune suppression that they need to prevent rejection. And those patients, well, depend on dialysis for the rest of their lives. That's an extremely intensive treatment. 3 times per week on average, 4 hours per treatment, sitting in a chair or laying in bed connected to the machine, feeling bad prior to the dialysis and feeling bad after. So it's just a way to stay alive. And the vascular access related complications are an important downside of the treatment. So about 25% of all hospital admissions of these patients relate to their access, so not to their high blood pressure or the technical problems with the machine or the treatment, but just to the access of the dialysis. And the current options, well, for some patients are suitable and functional, but with a very high need for interventions to maintain or restore patency of these conduits. The currently preferred type of vascular access for dialysis is an arteriovenous fistula, as depicted here at the left side of the arm, where there's a direct surgical connection between a artery, in this case the radial artery in the wrist, and in this case the cephalic vein. And that is done to create a high flow circuit, which is needed to purify a substantial amount of blood per minute at the same time, because otherwise you would need dialysis for a much, much longer time, and it's already 12 hours per week to end up with a kidney function, if I can translate that, to approximately 10% of normal. So even if you do dialysis 3 times per week, 4 hours per session, your kidney is only being replaced by approximately 10%. So that's why the high flow is needed. And in addition, because of the arteriovenous connection, and the higher pressure that's then being exposed to the vein, ideally the vein expands in size. So, and that's the process what we call outward remodeling. So, and it has some overlap with aneurysm formation in the artery, but here you actually would like to see growth of the venous outflow tract in order to cannulate the access for dialysis, right? Because you need to insert quite large needles to get a sufficient blood flow to the dialysis machine and back to the patients afterwards. And the current standard sort of blood flow for dialysis in the range of 350 mL per minute. And so that's why an arteriovenous connection is being made. It was actually a technique that, first described in the 1960s, also in the New England. And in those days, the average age of dialysis patients, because of the shortage of dialysis, was much lower than in the current time. Because that's when these kind of radiocephalic fistulas were first being made. The average age of the patient was, let's say, 40 or even lower. And now the average age of patients is 65 plus. Where these patients are much more obese, have much more vascular complications. So the outcomes of this type of access is far worse compared to the original publications. In patients where there are so many vascular abnormalities already present at time of start of dialysis, sometimes a direct connection cannot be made because the vessels are too small or too calcified. And then sometimes an arteriovenous graft is being placed in between the artery and the vein. So that's a polymeric graft made of polytetrafluoroethylene in between the artery and the vein, and then the graft is being cannulated. And the diameter of those grafts is usually approximately 6 millimeters, or sometimes a little bit larger. So that's the second option, which in the past in the US was the most prevalent types of access. Now it's getting more a bit towards the arteriovenous fistulas, because these grafts have a very high rate of interventions needed to maintain or restore patency. On average, about 2 surgical procedures per year are needed to be able to use that access for dialysis. For urgent dialysis, which we won't talk about for today, or patients with a very poor prognosis, a central venous catheter is being chosen for access. And the advantages of those is that you can immediately use them for dialysis, so 15 minutes later. But of course, because there's an open connection between the outside world and the vasculature, have a higher risk of infection, but also a risk of thrombosis because of the plastic that is inserted in the vasculature, causing adhesion of thrombocytes and subsequently thrombosis of the vessels in which these catheters are being inserted. So by saying that, I hope I can convince everybody that there's an urgent need for innovative strategies to improve outcomes of vascular access. And I know some of the people in the audience are working on that, and I would highly encourage you to continue with that. So this is what I already answered. We can be short on that. And the 1-year primary patency rate, which means the percentage of accesses that can still be used at 1 year without an additional surgical or a procedure. Well, the answer is that it is approximately 50%. And I'll show you that in the next slide. I'd like to speed up a little bit. And so you can have upper arm AVFs, and lower arm AVFs where the diameters are a bit smaller, and arteriovenous grafts in sort of gray. And you can see on the left side that 12 months, 50% of these accesses, especially for the lower arm AVFs, lost their primary patency. So the patients needed to be reoperated, or a percutaneous balloon angioplasty being performed. And around those interventions, dialysis is less efficient, need to be postponed, so it has a major impact for patients, but not solely for the patient side. It's also a huge burden economically for society, because in the US, for instance, I think that 12% of the total budget for Medicare is being spent on vascular access for dialysis, and it is about $3 billion per year. being spent on vascular access related procedures. So that's really, really massive. The type of lesions that occur, or the problem, is a bit different between AVFs and AV grafts. In AVFs, so where there's a direct connection between the artery and the vein, non-maturation is the biggest problem. So the inability of the venous segment to expand, to facilitate an increase in flow, because the venous segment is the segment that's being cannulated, and that's because it's just too risky to cannulate the artery multiple times per week, and the diameter of the artery is still too small because the artery does not expand as much as the vein does. While in the arteriovenous graft, non-maturation is not really the issue because the prosthetic graft is already at a proper size to do cannulation. So you can, on a relatively short term after the surgical implantation of the graft, use the graft for cannulation. But the problem is primarily at the graft vein anastomosis, stenotic lesions occur in the downstream vein, but also in the perianastomotic region. And there are multiple factors responsible for that. It's the compliance mismatch between the graft and the vein, but it's also the turbulent flow that occurs there, and sort of the strain injury, the repetitive strain injury that's being exposed to the vein, stimulating the cells of the vein to proliferate and to cause stenotic lesions.
Arvin, I see you.
Question. Yeah.
Yes.
Yes, so my question is, this 50% patency and stenosis, is it consistent across the different types of graft, or is it also like, if you have tissue engineered graft that's say aligned with endothelial cells, would you have or expect lower chances of it getting kind of stenosed, or is this just a general what's synthetic graft only, or can you comment on that?
Yeah, that's a very good question. I like that. The 50% is from the conventional ePTFE grafts that are used most widely in clinical practice. And of course, the idea of using tissue engineered blood vessels to replace these prosthetic grafts is that by getting rid of these prosthetic materials, which could cause an inflammatory response, that the outcome would be better. And theoretically, ideally, you would like to implant endothelialized grafts, because of course that could further improve the interaction with smooth muscle cells and keep them in a quiescent state. So that is indeed the rationale of using tissue engineered blood vessels. The question is, however, if for this specific application, if the studies that have been performed thus far really showed superiority compared to the conventional prosthetic grafts, or not, and I'll discuss that in the next slides. And if not, why not, and what can we learn about that? It could be very, very different, and I want to explicitly state that for the application, for instance, for peripheral arterial disease. If you make a bypass graft in the femoral artery, for instance, because the flow conditions are completely different over there, are much, much lower. There is not a very steep pressure gradient because the pressure is constant throughout the graft. These grafts are then not being cannulated because that's, of course, one of the major ways to ruin blood vessels, right? Putting very large needles in 3 times per week. And most people that work on vascular tissue engineering primarily look for the application as dialysis access, because if the graft occludes, the patient is not immediately suffering from severe complication, right? So they're not bleeding to death, so to say. If, for instance, if you put in tissue engineered blood vessel in a arterial, as a replacement of the aorta, if you then see a rupture, the patient dies, right? And here the blood vessel occludes, or you get bleeding complications, but because of the location in the arm, those bleedings are usually manageable. And I won't say that it's okay, but that's one reason. And the second reason is that, of course, the clinical need is very high for dialysis access because of these poor performance of prosthetic grafts. But at the same time, these patients are very, very sick, right? They don't have kidney function, only for usually. They have very calcified vessels, they're severely diseased, and you cannulate them, and the hemodynamics in these grafts are extremely challenging and very with excessive flow values and flow turbulence. So if a tissue engineered blood vessel is not perfectly functioning in a arteriovenous graft setting, we should not state that these blood vessels are not useful. It might well be that they function much better in a peripheral bypass setting. Merle.
Yes, so I guess this is also something you will talk about more, but to what extent is there evidence that this microenvironment, like calcification, like altered hemodynamics, like raised uremic toxins, has a detrimental effect on either remodeling or maturation of these blood vessels?
Yeah, good one. Well, I've been doing quite some animal studies from mice to pigs to goats, and in most of the studies we decided to do these, even though it was primarily meant for dialysis access, not in uremic animals. And every time I get back from reviewers, well, this is useless because your animals are healthy, and in the conditions of uremia, it might be very different. And I do think to some extent they are right. But if you look closely to the type of lesions that develop, for instance, in uremic conditions, morphologically, they look exactly the same. It's just that in uremic conditions, these lesions develop faster. And it might be a little bit that some of these growth factors in the surrounding are a bit more higher expressed than in non-uremic conditions. But in animal models, at least, whether or not they're uremic or have normal function, morphologically, I don't think there's any pathologist who could distinguish a lesion that was developed in uremic animals from a lesion that was developed in animals with a normal kidney function. So for these preclinical studies, I think adding uremia could accelerate the development of lesions, but it could also lead to a far higher dropout rate of animals, right? Because if you really want to mimic these conditions of humans, you really have to induce very severe kidney function. And if you want to really do the ultimate experiment, you need to dialyze these animals. And from an ethical and practical experiment side, I feel that is not needed per se to get an idea about the functionality of these grafts. By saying so, I fully agree that lesions in humans, for instance in the artery, the calcification in patients with uremia is substantially different from calcifications in patients with conventional or classical atherosclerosis. For instance, the calcifications are primarily located in the media in patients with uremia, and that's due to the sort of the tendency of fibroblasts and smooth muscle cells to calcify due to the circulating uremic toxins and the disturbed phosphate and calcium metabolism in these humans. While in the conventional atherosclerosis, the calcifications primarily occurred in the intima. So that's indeed, well, still I think a struggle in how to tackle this, right? And how to what extent animal models that we use do really have to mimic exactly the conditions where your grafts are being used for in patients. Yeah?
Yes, thank you.
Okay, so these tissue-engineered blood vessels, as I already mentioned, could be an alternative for dialysis access because there are multiple advantages, at least theoretical, right? Because they can be made with tailor-made dimensions. We can get rid of the problem of maturation, because if you implant a vascular graft of 6 millimeters, like a prosthetic graft, then you don't need maturation, and you can almost immediately after these anastomoses are being healed, cannulate these grafts. You don't need prosthetic materials if you choose to use purely biological tissues. And importantly, as I already mentioned, you don't have these vascular pathology that is the result of chronic kidney injury or chronic kidney disease, right? The calcifications and the endothelial damage and so on. And I just want to go through quite some examples of tissue engineered blood vessels that have been used in the past. And one of the first one is the use of genogenic decellularized bovine ureters. So no blood vessels, but ureters have been used by Eric Chemla from the UK and published already 13 years ago, where these ureters from these cows were decellularized and then implanted to be used as a dialysis access. But as you can see, the patency of these grafts was actually lower than conventional prosthetic grafts depicted in blue. And there were multiple options that could explain that. For instance, the residual xenoantigens that might be present in the tissue, or of course the absence of endothelial cells. The latter is less likely, in my opinion, because of course these prosthetic grafts are not endothelialized either. But anyhow, there was an increased risk of early thrombotic events in these ureters. So there was not being pursued. And with this publication, I think the application of bovine ureters stopped. Another example is also the use of bovine materials, in this case from mesenteric veins, which were decellularized prior to the implantation as a dialysis access. And again, it was quite a large study in 180 patients on chronic hemodialysis, which already had a failed prosthetic graft. So it was shown that this prosthetic graft had poor outcomes, but these tissue engineered blood vessels unfortunately did not perform better. So another disappointment. And then I'd like to discuss the work from Nicolas Leroux, who's currently working in Bordeaux, hosting one of the previous events of the ISACB. So probably some of you have met him already. Before he worked in Bordeaux, he was really still very innovative, but he was working in a company that was developing tissue engineered blood vessels, also primarily for dialysis access. And he used a very clever approach, where the idea was that they made blood vessels without a scaffold. So by isolating vascular cells from small vessels in the subcutis, by doing punctures of these subcutaneous fat, and they isolated small vessels from that, cultured endothelial cells, fibroblasts, and as well as smooth muscle cells, and with a very delicate technique, sort of peeled off these sheets of cells that were grown in these flasks, and they were wrapped around a temporary mandrel, depicted in green in my slide here. And then after the mandrel was removed, these layer of endothelial cells were applied to the inside of these constructs. And then these constructs were implanted in this case, I think it was also code of 10 dialysis patients. And here you see an example of the procedure where, as you can see, the tissue, well, looks quite like a blood vessel, at least microscopically. One of the downsides was that the processing time, at least in the beginning, was somewhere between 6 and 9 months. And you can imagine that it's also quite costly to make sure that you don't get infections and so on, because if the material get infections, you are not allowed or not able to implant it. But the burst pressure was quite high, it was above 3,000 millimeters mercury, so suitable for implantation. And in the report of in the New England, which was published in 2007, the primary patency of these grafts was approximately 60% at 6 months. So, which is actually exactly the same as prosthetic grafts. And the number, which was the primary patency, right? So not the secondary patency, so the cumulative patency, so to say. But at 6 months, in 40% of the patients, an intervention was needed to maintain or restore patency. So this study showed that it's really possible to implant these grafts in patients, and that you could avoid using prosthetic materials. But it did not really result in a broad application, because of course the patency was not substantially higher than that of prosthetic grafts. Another downside is of course the long procedure, the long time to produce these grafts, and the high costs associated with that. And for those of you who are interested, I think in the latest issue of the Journal of Vascular Access last month, they reported about the even long-term patency of well, up to 8 years or so of these grafts, so that's worthwhile reading. The most attention in the last decade or so, the work of Unicyde got the most attention, and that's another way of they developed another way of making in vitro engineered blood vessels. And their concept is that they have a sort of a donor bank of vascular cells, and that they apply to a scaffold that is in the bioreactor, as being put on the right upper panel. And those cells, they produce the matrix, and the scaffold is being resolved by itself. It's a biodegradable scaffold. And subsequently, because of these cells are allogenic, decellularization steps is being performed, because of course that would induce a immunological response, because these cells are not from the recipient. So what's left over is sort of a extracellular matrix made of human material. So that's sort of the scaffold that you all selected in one of the previous questions I raised. And then implanted as a arteriovenous graft or as a peripheral bypass graft. And they did a lot of preclinical studies, and was pioneered by Laura Nicholson from Yale University. And I think it took almost 15 to 20 years of preclinical development before the first clinical trials could be performed. And well, that's a major achievement. And you can see here, and these are baboon studies of 6 months, where on the left you see the decellularized scaffold that after implantation sort of is recellularized by autologous cells of the recipient, in this case a baboon. And that you could also see that in these, I have to say, healthy animals, an endothelial layer was observed at 6 months on the surface of these constructs. Merle.
Yes, I was wondering if you know, if we can come up with a hypothesis why some of these grafts, in the case of the human side, it looks like they get completely recellularized. So really cells infiltrate into this extracellular matrix, but other constructs, including your rods, that will more function like a scaffold themselves, and that then the cells will grow on top, but maybe less into the matrix. What would be the difference here? Because of course the ECM is not completely the same, but what would be the trigger that in some cases cells do infiltrate and form really a vascular construct within the scaffold, as to opposed to more on top? It's really interested to why this difference.
Yeah, that's a very good question. And I have to say, I don't have the clear answer, but here, because I will show the work that we've been doing on this in the next slides. But here, what's implanted is the scaffold, as you can see on the left, right? And I do think that, of course, the porosity of the scaffold, so as part of the structure of the scaffold, is very critical. And it's also, well, we have to, I don't think it's very well known from which side these cells enter the scaffold, right? If they end up from the lumen or from the sort of adventitial side. And what is known is that, for instance, and that has been described by us, but also by others, that, for instance, macrophages that infiltrate these constructs can differentiate themselves into fibroblasts and contribute to the fibroblast smooth muscle cell components of cells that end up in the graft. The question is, of course, how to steer that response of recellularization. And I don't, as far as I know, I don't think it's known in detail how to optimize the ingrowth of cells, and not solely because it's not solely about cellularization, because as you can see here, especially on the right side, the cellular structure looks still a bit well, I don't want to be offensive, but it's not very well organized, right? You don't see cells really aligned in a circular way. But for dialysis access, again, the question is if that's needed, because their contraction of these cells is not a critical characteristic. So, but here, I think, to coming back to your question, this is an area where a lot of work still needs to be done to fully understand the biology behind recellularization of these grafts. And it's a balance, of course. So if you make these scaffolds more porous, then you could probably facilitate the ingrowth of cells, but at the same time, the downside is that you lose mechanical strength, right? So that that's the challenge, I think, to find the optimal scaffold, is that it facilitates the biointegration and at the same time provides sufficient strength to prevent mechanical disasters or bleedings or ruptures. So I feel that that's one of the challenges that this field is still facing. But if there are other people around in the audience that have different ideas or have other knowledge to share on this topic, that would be interesting. So already 6 years ago in the Lancet, this first trial was being published, and probably some of you have seen this before, where these grafts are being implanted in dialysis patients, both from Poland and in the US, so in total 60 patients. And you can see on the right, you can see that also in humans, a recellularization occurs. That there is some variation in recellularization between grafts, and that in this case, the primary patency at 12 months in the Polish cohort was 33%, and in the US cohort was 13%. While the secondary patency, which is actually sort of the patency including interventions to maintain or restore patency, is much, much better. It's almost all grafts were still functioning, but as I just mentioned, interventions were needed. You can see also in this figure from the same paper that on average 1.9 interventions were needed to maintain or restore patency. And again, that is a similar rate of interventions compared to conventional prosthetic grafts that you can just buy off the shelf and being produced. That they're still almost €1,000 or $1 to buy, but production costs, of course, are much, much lower. So it's not solely about showing superiority, but also in a way that the production of these grafts, that the additional costs are in balance with the benefit compared to the current golden standard. And of course, that's the challenge, not solely for vascular tissue engineering, but for all kinds of medical innovations in general. And I always state, and I think that I might be right, that the majority of the innovative treatments that have been developed in the past always make healthcare more expensive. And of course, they have sometimes very high added value, and that's the reason then why it's good that we can use it. But the challenge is that, and that's for a debate in Europe and probably also in the US, although finance structures are much different, is that we don't want the healthcare cost in total, we don't want it to grow because we already spend so much money on healthcare, and I'll come back to that at the end of my presentation as well. And at the same time, we develop very expensive treatments that we all want to use, and that's some sort of a dilemma where no one has a solution for it. But I state it because I think in thinking about methods to, in this case, create tissue-engineered grafts, if you anticipate that these grafts will cost 30,000 euros each, the chance of getting it implemented in daily practice is very, very low. This is another method performed by Bob Tranquillo, and I saw him in the audience today. So hi, Bob. This is a good option to say hello again. It's time to meet in person. And Bob used fibroblast and entrapped in fibrin gels, which were decellularized before vascular grafting. And he showed sufficient burst pressure and suture retention strength. And you can see here in baboons that these grafts were also implanted as arteriovenous graft with very good and promising results. Again, at 6 months in this baboon model, similar as the model that was utilized by Laura Nicholson from Humanside. Let's see if we can. And here you can see indeed the explantations, how these grafts looked, to the that they also cellularize, and that there is some loss of patency. But again, here the challenge is to, as far as I know in this paper, but Bob, correct me if I'm wrong, there was no comparison with prosthetic grafts. So it's a bit difficult based on that to say whether or not the functionality is higher or lower compared to the golden standard. But you could say, well, at half a year, the patency is in the range of, well, it could well be that it's better than prosthetic grafts, so that we're looking forward to human results with these grafts. Then I would like to shift to the work that we've been doing ourselves here in Leiden for about 10 years or so, where we had the aim not to grow blood vessels in vitro, but to use the foreign body response to implanted materials to grow autologous blood vessels. And then indeed autologous blood vessels that were already cellularized with autologous cells prior to implantation in the vasculature. And as you all know, especially the ones with biological background, that the implantation of foreign material in the human body always causes a foreign body response, and the type of response and the extensiveness of the response to some extent depends on the type of material that you implant. So with selection of the implant material, you could actually steer that response. And most work in the past was being done, for instance, if you look to the applications of pacemakers or other implants, to use materials that are biocompatible as much as compatible as possible, so that the foreign body response is as small as possible. But for this purpose, of course, we needed polymeric materials that induced a foreign body response. While upon removal of that foreign material, the tissue capsule that's formed around it is being used for vascular grafting. Initially in the arterial circulation as a carotid interposition graft, and later on as a arteriovenous graft for dialysis. So this is an overview of the very dynamic foreign body response, where a lot of proteins and cells are involved in the initial phase to induce the primarily inflammatory response, where initially neutrophils bind to the implant material, and later on the macrophages take over that response. And even further more down the road, you primarily see fibroblasts in these tissue capsules together with collagen. And then if you wait even longer, then most of these cells, these fibroblasts, die as well, or at least leave the tissue capsule. And then what's sort of left over is primarily scar tissue. So it means that the timing of removal of these rods or foreign materials is critical for the composition of the tissue that you subsequently are grafting in the vasculature. So it's the material itself as well as the timing of the development of the tissue capsules. So this is an overview of the concept that we worked on. That's where in the past I got grants for to work on, where we actually, the idea would be is to implant the polymeric rod at the site where the vascular graft is needed, so that the tissue capsule that is formed around the rod, in this case, so the polymeric rod, could be kept in place and that upon removal of the rod, you just anastomose both ends of the tissue capsules to the artery and to the vein, with the advantage of the perfusion of the tissue, right? Because it's then perfused by the surrounding tissue around the tissue capsule, and that you then can cannulate these tissue capsules for dialysis. And when I was thinking about it, I actually realized that there's, I found out by reading the literature that that has been tried in the past, and the elderly people in the audience probably are aware of the work of Sparks. Charles Sparks was a vascular surgeon from the US, who already in the 1970s used some sort of approach where he implanted materials, in this case in the leg of patients, with the aim to induce a foreign body response around that implant material. And here you can see some examples of the tissue that was formed around the implants that are depicted on this slide. That method was stopped because of the very poor outcomes, which related to aneurysm formation as well as thrombosis. And of course, well, when I found out, we asked ourselves, well, should we stop this, or what actually the primary reason why these outcomes are so poor? And one of the ideas would be is that, of course, at that time there was no knowledge at all about, or not, that's a bit too strong, limited knowledge about the foreign body response and the type of materials that could be used. And as you can see here on the histology, you could see that the cellular density in the tissue that was grafted was very, very low, and that at the same time there were some polymeric material still incorporated in the tissue capsule, or in the tissue capsule that was formed around. And those two things could well be the primary reason why the mechanics of these tissue capsules on the longer term are just not sufficient to prevent aneurysm formation and thrombosis. So we first spent a couple of years on optimizing the material. We used a copolymer with the experienced help of Lorenzo Moroni, who's a professor of biomaterials in Maastricht University here in the Netherlands. And we subsequently modified the surface of these polymeric rods with all kinds of chemical etching or plasma treatments, or a wide range of modifications. And we then looked at the surface because the surface, to some extent, of these polymeric rods determines how easy it is for cells to attach, and to some extent also the aggressiveness of the tissue capsule that's formed around it. So we first did studies to see at the, let me see if I can, here you can see, for instance, in vitro, with co-culture with cells, that we could observe that the number of cells that were bound to the material were strongly dependent on the different type of surface modifications that we performed, and that also, if you look on a closer level on these on the collagen synthesis in these cells, that substantially differed between these surface modifications. So we then implanted a variety of these polymeric rods in rat, in the subcutis of rats, and here you can see an example where we had just an unmodified polymeric rod implanted in the subcutis, chloroform etched rods, and rods that were coated with TGF-beta. And we then primarily morphologically examined how these tissue capsule looked. And you can see that here in the middle panel with the chloroform etching, that the collagen layer looks much thicker than in the unmodified rods that are more alpha-actin positive. In this case, myofibroblasts are present in the tissue capsules, while the TGF-beta coating actually accelerated the tissue capsule formation even further, but in a bit more disorganized way. So the collagen was not so nicely aligned. So we decided to continue with the chloroform etching, also because that's a modification that we could do in a very standardized way. We also did studies in, on the topic that we already discussed, in uremic animals. So these are rats with 5-6 nephrectomy, which is challenging, of course, because these animals have a very poor condition, and that causes additional burden to the animals. But we felt also for the clinical application that it's quite critical to know whether or not that foreign body response is different in uremic conditions. And we found out that that was actually not the case, that the tissue capsules were also formed in a similar way in these uremic animals. And then Taya Besheva, former PhD student in our group, performed very elegant studies in collaboration with the University of Los Angeles to find out actually the origin of these cells, because we observed that indeed quite strong tissue capsules were formed and consistent, but the question is indeed if these cells originate from the bone marrow or from the surrounding tissue. And so she used GFP labeled bone marrow cells obtained from GFP rats, which were, and she did a bone marrow transplantation in these rats that also got a subtotal nephrectomy, so with a 5-6 nephrectomy model. And then we checked the histology of the tissue capsules that were formed around these rods to see, again, as I already mentioned, if CKD would affect tissue capsule growth and also where these cells were coming from. And she observed two major observations. One is that only a small proportion of these cells that ended up in a tissue capsule were bone marrow derived. And secondly, that a large proportion of the cells that expressed alpha SMA also expressed CD68, suggestive of differentiation of macrophage towards a myofibroblast type of cell. And that was actually already being described before, primarily by the group of Julie Campbell in Australia, and that's actually the group where I started this work together with Julie Campbell, where she used more like an intraperitoneal approach, where she implanted polymeric rods in the peritoneal cavity to induce tissue capsule formation around these rods. But it learned indeed that these macrophages, which initially are very inflammatory, actually do have the capacity to transdifferentiate into a fibroblast-like cell. And that is what most tissue engineering studies actually show, that the initial phase of invasion of macrophages, that it could well be that the same cells actually later on are still present in the tissue, but then in a less inflammatory status as fibroblasts. As I mentioned before, mechanical characteristics are quite pivotal. Here you see an example of a tissue capsule that's grown in a pig, where we actually increase the pressure and we measure the pressure, and you can see on the right the pressure that was measured. And in this case, the burst pressure of these tissue engineered blood vessels was about 2,500 millimeters mercury. Well, we compared that to carotid arteries of the same animal, in this case a pig, and the burst pressure was a little bit lower than the carotid artery, but still high enough to allow safe implantation. And that's actually what we first did in this case in a pig model of carotid interposition grafting. I have to say, this is a bit it's difficult to distinguish it here. But this is actually the tissue capsule after removal, 4 weeks after grafting, so a short follow-up. And that we intentionally choose that because we know that in these animals, in pigs, it's extremely challenging to, even if we would transplant a carotid artery from a pig to the other side, then still you would see the formation of stenotic lesions. And so that's why we choose a small, short follow-up time. And we observed that the majority of grafts were still patent after 4 weeks, but that indeed also at the anastomotic size, some stenosis developed, but that we could cannulate these grafts and that the burst pressure remained stable. So we repeated these experiments of these mechanical testings after 4 weeks, and we did not observe a change in burst pressure, which was of course good, because that could be the case. And then we moved on to a goat model of arteriovenous grafting. Here you can see an example of a video that we made of one of these procedures, where we used the goat also because the neck of a goat is a bit longer, as you can imagine, than from pigs. And so there's more space to create these arteriovenous grafts. So here we just insert the polymeric rods, And then 4 weeks later, we first optimized the incubation time, so to say. So we did 2 weeks, 4 weeks, 8 weeks, and so on. But at 4 weeks, we observed that the tissue capsule already looked strong enough, and the composition looked good to really graft that tissue capsule as arteriovenous grafts. So here we remove the polymeric rod with its surrounding tissue, which, as I mentioned before, is different from the ultimate way of working for the human application, because there we want to keep the tissue capsule in place. But because the carotid and artery and the jugular vein in goats are laying much deeper, we had to dissect the tissue and to graft it actually between the artery and the vein. And that's what you can see here. You see the very turbulent flow that is so typical of arteriovenous grafts and fistulas, and tissue that, well, at least macroscopically, looks quite a lot, looks similar to blood vessels. Here you see some examples of electron microscopy analysis of the tissue capsules depicted on the right. Well, after grafting, in this case 4 weeks after grafting, we then call them tissue engineered blood vessels, because of course before grafting they have not been in the circulation, and it's a tissue capsule formed around the polymeric rod. But we observed that indeed the surface area of these tissue capsules change upon implantation in the vasculature, and that there's a sort of a monolayer of cells on top of it after it has been implanted for 4 weeks in the as arteriovenous graft. You can see that these prosthetic grafts of the PTFE graft in the left lower panel, there's no cell attachment at all to it. While of course the nature is always the best. The carotid artery looks very nice with a very flattened monolayer of endothelial cells on the surface. You can see in tissue capsules that there are a lot of cells, including thrombocytes and inflammatory cells on the surface. Right? But if you implant it in the carotid artery or as an arteriovenous graft, the flow is so high and the diameter of the vessels is so big that it does not result in acute thrombosis or occlusion of these grafts. And it's, well, I don't have the time to discuss, although we have a lot of time, but not the time to really discuss in detail the role of platelets and so on in the vascular healing. But well, we do feel actually that the adherence of platelets here could actually help in the recruitment of endothelial progenitor cells to stimulate endothelialization of these tissue capsules. What we then did is we did some sort of a thrombogenicity assay. So we built a device, as you can see here, where we put in this device a prosthetic graft, part of a tissue capsule, or the tissue that have been grafted 4 weeks ago, and then perfused it with uncoagulated blood of the same animal. And you can see here again is the different examples. So the conventional artery, carotid artery of, in this case, a goat, the tissue capsule, the tissue engineered blood vessel, and a PTFE graft. And you can see in the unperfused setting on the left, well, the differences between them. And you can also see that, for instance, if you perfuse the tissue capsule ex vivo with not anticoagulated blood of the goat, that you get massive adhesion of thrombocytes and fibrin and so on. But of course, this is an ex vivo experiment. In vivo, this does not translate into complete occlusion. And if you repeat that with the tissue that have been in place for 4 weeks as an arteriovenous graft, then hardly any thrombocytes and platelets adhere anymore. So the surface has changed in time in these, again, I have to say, healthy goats. And, well, we try to quantify the adhesion, and you can see that in the tissue capsules actually completely covered with platelets and fibrin, while the tissue engineered blood vessels, the percentage of adhesion is, well, very close to normal arteries from these same animals. Well, then to the histology. As you can see here, for instance, on, well, just conventional HBS staining, MOVADs and collagen stainings, and also stainings for inflammatory cells. This is, I think, a CD68 staining on the right. And you can see that actually the collagen content is quite stable over time. It's a little different than the carotid artery, but the density of the collagen seems to be quite close to conventional arteries. But if you look, for instance, to the number of inflammatory cells, so the tissue capsule at 4 weeks after implantation of the polymer rod in the subcutis still contains quite some inflammatory cells. After 4 weeks, that number is reduced, and after 8 weeks, we could hardly detect any inflammatory cells in these tissue capsules anymore. So that because a foreign body is no longer present, there's no real additional stimulus for inflammatory cells to invade in these tissue capsules. And also, if you look to smooth muscle cell characteristics, you can see here, for instance, that if you look to myosin heavy chain, which is a marker of really contractile smooth muscle cells, you can see that in the carotid artery in the upper panel, most of the cells are positive. In the tissue capsules, we don't see any myosin heavy chain at all. But then in time, the percentage of cells that starts to express myosin heavy chain increases, which again is suggestive of some sort of a transdifferentiation of cells of these fibroblasts that starts to act like smooth muscle cells. And of course, it's always a debate whether or not you could use these type of markers as functional readouts of these cells, because the fact that they express contractile proteins is not the same as functional contractility. So what we try to do is to do a contractility assay where we isolated the cells from these tissue capsules and tissue engineered blood vessels, as well as from the artery, and we used fibroblast as a control. And you can actually see here with this contractility assay that in these tissue capsules, so before grafting in the arterial circulation, there was hardly any contraction of these cells, because you use the deformation of the collagen scaffold, or collagen sort of layer in which they are cultured. If these cells contract, you can see a change in configuration of these of the collagen. And after grafting, so when we color the tissue engineered blood vessels, there's an increase in collagen deformation, suggestive of indeed not solely the contractile protein expression is increased, but that these cells indeed have more capacity to contract. We also looked, of course, at endothelialization and observed, again, in these healthy animals that these tissue capsules were nicely aligned in time with a monolayer of, in this case, CD31 positive cells. And then we looked to the outcomes. We did 4 weeks and 8 weeks follow-up studies. The numbers were unfortunately a bit small, but we observed that at least there's a tendency of improved outcome compared to ePTFE graft. So as you can see here, at 8 weeks after grafting, primary patency of ePTFE arteriovenous grafts in goats is solely 33%, which is in line with literature from other groups. So, and there, of course, that's the debate that I tried to start as well. If we would have here selected a follow-up of, let's say, a year, it's almost impossible, because all PTFE grafts will be occluded after 6 months. And because of these excessive hemodynamic conditions, I don't think that any tissue engineered blood vessel will ever show functional patency at 1 year in this goat model. And then if you select yourself that as an endpoint, your therapy would fail, unless it might still be very suitable grafts for clinical application. So then it's time for me to shut my mouth for a while and ask you a question, because I noticed that the majority of the participants is from the US, but I think there are some similarities about in terms of regulations. And in the European Union, the medical device regulation has been implemented recently, which changed a bit the way we have to deal with preclinical and clinical evaluation of medical devices. And so the question here is this a class 1, class 2, or class 3 medical device? And I mean then the polymeric rod, right? Because that's the implant, which would stay in place for about 4 weeks. And the good answer here is class 3, because it's a medical implant that is indeed that stays in place for, in this case, about a month. And the classification is of importance because it guides, well, researchers and people that work on medical innovation, how the assessment and the evaluation, preclinical as well as clinical, should be done. And in general, the MDR makes it more difficult, at least in Europe, to make the transformation towards clinical evaluation, because in the past it was primarily needed to show safety of a medical implant, and now there's more focus on efficacy. So you really have to show that the implant that you would like to introduce into clinical care does what it should do, which makes sense from my perspective. But it increased the hurdle and the cost, of course, to develop these kind of implants. So, and that's good to be aware of. So for class 2 and class 3 medical devices, you need to get in touch with a notified body who will discuss with you how the development plan of the medical implant should look like, and that you, in the US, you do that with together with the FDA, and that you make a plan for that whole, well, for the development plan and the proof that is needed to get approval for clinical studies, as well as later on for entrance at the market. So I don't think it's needed to discuss it very extensively, and already made some remark about it, but in general, the implementation of the MDR resulted also in a, well, far more extensive clinical evaluation of its performance, focus on efficacy instead of solely on safety. And that's good to be aware of. So, and one of the, when we were developing, or we were discussing this webinar a while ago, and the topics that we wanted to address, apart from research itself, well, one of the issues that we discussed was also, so why is it so difficult, right? Well, you see, so now and then these papers in these high impact journals are rising, showing very promising results of tissue engineered blood vessels or other medical technologies, but only a small proportion of them will be actually applied into patients. And well, and that is, I think, for multiple reasons. And one of them is, of course, the challenge in finance, because of these very strict regulations, it's quite expensive to really build a development program that meets all these requirements. And of course, it's not solely about research, right? So the questions there about product development and standardization and all these kinds of questions are different from the scientific questions that where it all started with, about well, the underlying biology, and really focus on the efficacy and so on. And in terms of finance, it's frequently the term valley of death is being brought up, because it's, and that's what I experienced myself as well, that if you would like to shift from the academic research towards really the making of a product for commercialization, or that should be on the market, you need a lot of money, not academic money, right? So you need to get in touch with investors and all kinds of funds that are available to set up spin-off companies and get some initial funding. And that's something you're aware of already when you are in the early phase, right? In which probably most of you are, if you are working on vascular tissue engineering in the academic phase. And if you, it's good to be aware of all these steps in the development plan, not solely about funding, but also in the development plan, because the more you do at the beginning in terms of, well, that are in line with the MDR, the less work needs to be done later on. And well, this is the topic that I already mentioned, right? And that's, I think, also another challenge for these kind of innovative work, is that if you realize that healthcare costs in almost all countries in the Western world are still increasing massively. You can see it, in, for instance, in oncology, where the new immunotherapy provides a far better perspective to patients than in the past, but at the same time, the treatment per patient is about 100,000 euro, while in the past, of course, conventional chemotherapy was much, much cheaper. And we all want to have access to those therapies, but we don't want to pay more insurance for ourselves and for our fellow citizens. So that is something to, you could say, well, as researchers, that's not our job. And well, that is an argument, but at the same time, as a community or as all countries in the world, we need to find a solution for that, or we should, to some extent, at least think about innovations that are not so expensive as some of the previous ones that have been developed, and to think about the relation between industry and academia, and the same is true, for instance, as well. If you look to the economics of the vaccine development, right, for COVID, that is also a good example. So why are we not so successful in bringing these technologies to the patients? And that because it's extremely complex. That, for instance, if you are in a spin-off company developing these kind of blood vessels, the simple question, who's my customer, right? My customer, is that the doctor, is it the patient, is it the hospital, is it the health, is the insurance company? Who do I need to talk to? Who do I need to get involved? And this is just a figure to illustrate how many people and institutions and issues are being involved in that development. For you as researchers, at least I assume that most of you are in the academic field, it's also good to realize, and I have seen too many examples of some sort of mistakes in the past, that because we're all focused on writing papers, right? Our goal, if you're a PhD student, your goal is to get a PhD and to get a large PubMed list and to end up in high impact journals. But if you are working on innovative technologies that with the ultimate goal to reach the patients, it's good to be aware that patents might be as important or even more important to apply for next to your publications. And that's the wrong direction. So my question, just to get an idea from you, would be, during my research project, I'm in contact with our technology transfer offers who can actually help with you for the patent applications. I know who to ask, or I know that they are there, or yeah, well, it looks like about a 50-50. And I'm not saying that the ones that answer here no are wrong, because that they should have. Of course, it depends a bit on the type of research and that you are doing, whether or not it makes sense. But I just wanted to make the general remark, and that's also a task of supervisors, of course, to be aware that if you think that some results that you obtain or a technology that you're working on has the potential to be applied in humans or to be part of a product for patients, that it's good to discuss prior to publication whether or not the application in a patent is worthwhile. Because for spin-off companies and larger medical industries as well, if there is no protection to the technology, then the chance of ending up in patients as a product or a therapy are very, very small. Also for yourself as a researcher, and that's what I experienced myself as well, if you as an academic doctor or researcher want to continue with the research that you did in the preclinical phase, and that you want to be part of the team that develops this product for human applications. It's good to be aware of the potential risk of conflict of interest, right? So suppose these blood vessels are available for patients, and you treat your own patients, and you prescribe your tissue-inhaled blood vessels to your patients where you are a shareholder of the company, right? And that you actually directly earn money from the treatment that you provide to your patients. Is that okay, or is that an unwanted situation? And it's quite remarkable that throughout the world, it really differs from country to country, what is allowed and what is not allowed. And also in terms of conferences, of course, well, these kind of scientific conferences can only take place with sponsors, but sometimes you now see more and more that sponsors are also involved in these sessions themselves. There's a more direct communication between researchers and sponsors. And I'm not saying that's a bad thing, but it's just good to be aware of this issue. And I think transparency here is the key word. Personally, I feel that researchers should be involved in the clinical evaluation, and that there are multiple ways of doing that properly, as long as you are transparent about the role that you have. And so I wanted to ask you what you think about that, and to bring that back to a question. The statement would be, it should be possible for academic researchers or clinicians to own shares of spin-off companies that develop their own products. All right, so everybody is saying here, yes. I don't have the question, but I would like to ask you, do you know if that is allowed in your country? Because, for instance, at my center, it's not allowed. If I wanted to participate in the spin-off company that is being set up, well, to some extent by myself, that is bringing the tissue-inhaled blood vessels to the clinic, I had to resign from my work as a academic researcher and a clinician here at the Leiden University Medical Center. And well, for me, but that's a bit of a personal story, because I'm a researcher and a doctor, and I like that very much. I decided not to give up my job here and to continue as a doctor and a researcher, and ask others to continue with the product in the clinical evaluation. And I know that, for instance, in the US, it's far more, I would say it's easier, but the rules are not so strict. And also for non-doctors, it's different in the Netherlands than for medical doctors. Well, it's too much of an extensive topic to discuss in detail, but I wanted to stress that it's good to be aware of potential conflict of interest. For the future, how to move on with this field, I don't want to make it too complicated, but there are so many things going on and so many new technologies that could be helpful to further improve outcomes of vascular tissue engineering. And even though this far in clinical evaluation, the golden solution has not been found yet, there are so many tools to further improve these blood vessels. And well, we all know that in research things go slow, and that we just should continue with our aim, our strategies to further improve outcomes. And I'm looking forward to reading all your publications, because probably many of you are working on these technologies to further improve the functionality. So that brings me to my two main conclusions, that there are multiple innovative strategies being developed for tissue-inhaled blood vessels, and that although I don't have a very clear answer, that in general, a focus on translational medicine is urgently needed to evaluate if these strategies could fulfill their potential to improve outcomes in vascular medicine and in vascular access for dialysis in particular. With that, I think it's time to shut my mouth and to see if there are questions from you, but not before. I would like to thank a lot of people depicted on the slide here who at least contribute to the work that I've presented from our own group. We collaborate with researchers from within the country, from Leiden and Maastricht, primarily on the biomaterials, and also from Utrecht, where all these large animal studies are being performed. And Boris Hintz from Toronto helped a lot with these assay on contractility. And of course, the funders from the research project from Leiden, from the Dutch Kidney Foundation, and also from the Dutch Organization for Scientific Research. Okay, thank you for your attention.
Thank you very much, Professor Rotmans, for this very clear and broad overview of all kinds of different approaches for the vascular tissue engineering field. I already have some questions via chat, and I think this is the perfect moment for people to come up with their questions and raise their hands. So I will start with one, or actually two, I got over the chat, so that people have some time to formulate their own questions. So this one is actually from Fabian Kawecki, who is from the Nicolas Leroux Lab, and he will be upcoming chair in the next webinar. And he said, "Dear Dr. Rotmans, thank you very much for your really inspiring presentation. I have two questions. One, is there a recommended time point for grafting of a tissue engineered blood vessel in a large animal model regarding the European Regulatory Agency requirements before going into human trials? And do you know if this differs from the FDA?" I'm sorry, I hope that was clear.
Yeah, thank you for the question. I think the short answer is no. It has not been defined strictly in the MDR, so what follow-up time is needed. And as far as I know, but maybe my American colleagues know even better, I would assume that also in the FDA regulations, it's not being defined so strictly. And then, of course, there are two ways of doing it, or you could go to them and ask them, so what is the interval that's needed, or you could think for yourself, well, I think this is the optimal interval or time point that is needed to convince myself and propose that as a part of your development plan to the authorities. And I would recommend to do the latter.
Yeah, okay, I think that's very clear. And I think, again, that maybe emphasizes on that you can defend for yourself that this is relevant. Okay.
That's in research in general the case, right? You first convince yourself that your results are true and then convince regulators.
The others, okay. And his second question was, what is the structural difference between your tissue capsule and the biotube vessel that is developed by Dr. Yamanaka of the biotube company?
Yeah, well, there are quite some similarities, and I think there are more similarities than differences. The main difference is the polymer, the implanted rod. So they have two different rods being used in the past. So I think a silicon rod and a metal rod. And we, based on the research that we preclinically did, screened a lot of different copolymers and have a specific copolymer that we developed for this purpose. Then the question is indeed, which is a difficult one, does it really matter what type of implant you use? I do think it does. Based on our preclinical studies, it does. To what extent that translates into differences in humans, I don't know, because there have not been very systematic comparisons being made between different implant materials.
Okay, very clear. Thank you. So I would like to invite the others to also share their questions, either in the chat or by raised hands. I haven't seen them yet, so I'm also looking at Arvin. In the meantime, I have a question that's maybe not so much content related, but of course we also have lots of early investigators here. And you mentioned that the tissue engineering field is extremely interdisciplinary or multidisciplinary, maybe a field more so than others. So can you maybe comment on what type of skills you are looking for in people that should want to work or continue to work in this field?
Yeah, sure. A very good one. Of course, well, they should motivated people, priority number one. Intelligent people, also quite helpful. But the most important is being a team player, right? Don't want to score yourself solely individually, but realize that the knowledge that you have is critical, but that you need others to really complete the whole route of development, and that you have an open mind in involving other people and trust them initially. Don't just start at day one about the distribution of authorships, but just try to first make friends and build trust, and then learn from each other that 1 and 1 is 3 if you find your good colleagues.
Yeah, great answer. I see, Arvin, you have a question?
Yes, I have a question. So first of all, a really great talk and really detailed presentation on the work that you do in your lab. One of the questions that I have is regarding your application for the tissue engineered blood vessels. I like the idea of repopulating it, having like endothelial cell formation and starting to get contractile cells in there. One of my question is, in one of the patency table, it seems like at 4 weeks, your grafted tissue engineered blood vessels 100% patency, and then at 8 weeks, it reduced to 66%. Do you know if that was happening between the 4 to 8 week period, or is it like something that happened early on? Because I expect until endothelial cell starts to form, there is a vulnerable period of where there might be more stenosis and maybe early clotting.
Yeah, a very good question, Arvin. It happens, it occurs somewhere between 4 and 8 weeks. And I agree that what we observed is that in the recipient vein, so at the site where the graft is anastomosed to the vein, in the vein, stenosis developed. And that was, well, to some extent quite frustrating, because even after 8 weeks, the tissue engineered grafts still looked perfect. There was some minor intimal hyperplasia, but not causing really severe stenosis. It was primarily in the vein to which the graft was adhered to. And that made us realize that it is the hemodynamic conditions that are so much of a stimulus in this model for the formation of stenotic lesions. And that also resulted in a new program that I didn't show today, but where we try to modulate flow conditions, right? Because it's such an unnatural environment of an arteriovenous graft of fistula that the way to prevent these venous lesions is not, at least in my opinion, the ultimate answer is not implanting a tissue in a blood vessel, is improving the hemodynamic conditions. Yeah, on the endothelium, of course, ideally, you would like to implant a completely endothelialized graft. And for instance, for small diameter applications like in coronary artery bypass grafting, I do think it's pivotal, because there the flow is low and the risk of acute thrombosis is relatively high. In these very high flow conditions as an arteriovenous graft, I'm not so sure if these endothelial cells are really needed. In the work of Humacyte as well, you don't, and their constructs, it is non-endothelialized, and almost all, except from Nicolas Lereu's work at the time of cytograft, these constructs, none of them are endothelialized. And you don't experience acute graft thrombosis, again, because of the high flow. The ones that in the past tried to endothelialize these constructs prior to implantation, most of those, and we tried ourselves as well, are unsuccessful, because directly after implantations, all these endothelial cells are just washed away because of these turbulent high flow. So it's a waste of time and effort, in my opinion. For this application as a dialysis access, and obviously it's different for other vascular applications.
And a follow-up question to that is how, in let's say hemodialysis patient that requires repeated needle puncture, how robust and strong is the engineered vascular graft for those kind of repeated kind of puncture exposure?
Yeah, well, we did not decide to include a dialysis procedure in these goats. So what we did is we did, punctures at time of harvest. And so that's not really repetitive punctioning. So we don't know. in all the other, for our specific grafts, for all the other tissue engineered grafts that have been used in dialysis patients, like the ones from Unicyte, repetitive cannulation seems not really to be the issue, because the healing response of this tissue is, well, definitely superior to the healing response of prosthetic grafts, right? Because there is just a hole ending up in the graft that's being filled with thrombocytes. So that, I don't think that's really a concern or an issue for most of the methods that have been used in patients.
Thank you.
I want to invite the other participants if they have any questions or remarks, either in the chat or with a raised hand. I don't see one at the moment. I think maybe I have a final question, or maybe Arvin has a final question. And after that, I would really like to show you a couple of closing slides, only two for the things that are coming up. So my question still is, you mentioned one of the improvements would be to improve hemodynamics, but we know for especially blood vessels and the smooth muscle cells that their maturation is enhanced by both the flow and stretch and strain. So for now, once these rods are implanted subcutaneously, these mechanical stimuli are of course very different from what they will encounter in the bloodstream. So would there be any advantage of still kind of preconditioning these grafts before they get implanted in vivo, or is that useless?
Yeah, very good one. That has been tried, and not per se with the tissue capsules obtained from subcutaneous implantation. But it's a group in Belgium that did that with other tissue engineered constructs that put on a perfusion pump prior to grafting to allow these cells to get used to these hemodynamic conditions. I'm not aware of any data in large animals showing that it indeed improves the outcome. But concept-wise, it really makes sense, right? But still, I agree that these the mechanical stimuli are the driving force for maturation or remodeling. But my statement is that in the arteriovenous construct as we currently use it, these conditions are just too excessive. Well, so to give you an idea, in the venous outflow tract, the flow is increased 40 to 50 times compared to normal. And there's a pressure gradient from 120 millimeters of mercury to almost 0 in a couple of centimeters. And that's just, I always say we shouldn't blame the vein for responding in the way they do, right? Because we just use them for something that they were not made for.
No, that's definitely also true. So I would like to ask if there's one closing question or remark about the presentation, about own experiences. And if not, I will try at least already to see if I can share my screen. Not yet, but I will be able to though. So yes. Okay, so yes. So before going to our next webinar, I think we should first take the time to thank Professor Rotmans for this presentation and this extensive overview, and of course for us, for Arvin and me, the opportunity to chair for the young investigators. And we think actually this is a super nice initiative, and we would really like to do this more often. So this is actually what we would call the pre-Memphis webinar, and we hope that with many of you we can follow up in Memphis in person. And then after that, we would like to continue with the post-Memphis webinar, which is for now planned in November. And we will have Professor Ellen Roche from MIT as our speaker, and Fabienne Kawaki, who was previously also here as the chair. So we will keep within the topic of vascular tissue engineering, and Professor Rotmans now more elucidated the bedside part, but also the difficulties in translation. And then Professor Ellen Roche will continue more from the bench point of view. So this is already an announcement that this is coming. So please block your agendas and yeah, be there as well. And if you like this initiative, we are still also looking for people to join our Young Investigators Committee, both from North America and from the EU. So if you are a young investigator or you know young investigators that would be interested, you can either contact Arvin directly. His email address is here. Ranjana Rai, who is from the TSUI lab in London, or myself. And we are, of course, mentored by people from the PIs from North America and EU. So if you know them, you can also try and reach out to them directly. So I think with this message, I would like to conclude the webinar for now. I would like to thank everybody for being here. Of course, this is going to be shared online. So I guess if you still have some questions afterwards, feel free to ask them. I think Professor Rotmans is more than happy to answer any questions afterwards as well via email or whichever other way. So thank you for being here. I will see if I can already stop sharing. And hope to see you guys in Memphis and otherwise in November.
Yeah, thank you as well. And sorry for I apologize for taking so much time. I now see how the time is.
It doesn't matter. I think there, especially for people, some people that have had to leave earlier, that they can look at the recording. So it's I think it's perfect like this. Okay, thank you very much, everybody.
Thank you so much.
Yeah, see you around, hopefully in Memphis. Bye.
Bye.
Bye.